Hi students, so this is a second class on cardiac muscle and this would be a very short class. I already have explained about Frank-Starling's law. Yes, I am. The previous class we stopped at Frank-Starling's law. Now we would go into this property. So which property were we actually discussing? We were actually discussing the distensibility of the heart, having been completed all the other properties. So in this context, we were discussing the Frank-Starling's law and the Starling's law, and you already learned that the force of contraction be directly proportional to the initial length of the muscle fiber within physiological limits. Okay, now what we are going to learn is about this length and tension relationship in the heart. So it's basically what we learned, but we have to learn in this context of length tension relationship in the heart, where we would be learning a graph about the interpretation of this particular graph. So let's start. This is what basically is called the length tension relationship. So what is basically length tension relationship? Famous Frank-Starling's law. Let me explain. I hope you have already understood what is preload here, written here. So preload is basically end diastolic volume. This would be the amount of blood which has come from the veins onto the right ventricle of the heart. So if this is the right ventricle and this is the right atria here, so blood has come into the right ventricle and this would stretch the muscles of the heart, and that is what is called preload for the heart. Now that so basically increases the initial length of the muscle fiber, and this is going to increase the force of contraction in physiological limits. Now let's go on to this graph and let us discuss the graph. And before that, as the end diastolic volume increases, there is an increase in stretch of the muscle fibers, there is initial length of muscle fibers, and as you know, there is increase in force of contraction. And this is your graph. This is the graph which shows the length tension relationship in cardiac muscle. So whenever you are asked to draw a graph which shows the length tension relationship, you will have to draw this graph. This graph has to be drawn. Let's discuss this graph in detail. So what you basically have to draw is are two axes. So this would be your x-axis and this would be your y-axis, and in y-axis you can see the pressure in millimeters of mercury, and on the x-axis you can see the diastolic volume. This is the end diastolic volume. You can't see the end diastolic volume because it's getting filled. So the scenario is this: the blood is as the blood comes onto your right ventricle, the ventricle will be filled with blood. First 10 ml is filled, 20 ml gets filled, 30 ml. So it increases slowly. It increases. If you draw it till 70 ml filled on your ventricle. So just think about right ventricle as of now. And it also happens in left ventricle also. This will happen when the oxygenated blood comes from the lungs and passes through the left atria and reaches the left ventricle. So let's now discuss this graph in terms of the right side of the heart, the right ventricle. So we have 70 ml of blood now here. Coming to the y-axis, you can see the pressure. The pressure that is built up inside the ventricles is shown in the y-axis. 0 millimeters of mercury to 270 millimeters of mercury. That is the pressure that develops inside. So now let's see the blue line that is shown here. How do you basically draw the graph? You draw from here. From this point you start. This is the starting point. You start here. And this experiment is also done in such a way that there is no blood in the heart and the blood is filling in the heart. So the diastolic part of the heart or the filling phase of the heart is first shown. So slowly blood comes and 10 ml is filled here, and now it's 20 ml filled. So as we see, as the blood comes and fills the ventricles, slowly you can see the pressure is rising, right? So you know this, when the blood is filling the heart, there should be pressure increase, even though the heart is stretching by itself, even though the volume is increasing, it's stretching, the muscles are stretching, there is a build up of pressure that slowly builds up. See? So the pressure at this point, if suppose we take the 50 mark, when 50 ml of blood is filled, the pressure would be 60 millimeters of pressure during the filling phase. That is during the filling phase. So all these pressures are during filling phase. During filling phase, the blood is filled 40 uh, 40 ml. The pressure in the ventricles during the filling phase would be 30 millimeters of mercury. So in this manner, it slowly fills up to 70 ml is shown here. And 70 ml fills. See, from here, from 50 to, from this point of 50 to 70, you can see that the pressure during the filling phase actually rises very fast, right? There's a sharp rise, declining phase. And when 70 ml is filled, the pressure that you can see here during the filling phase, 70 ml fills the heart, is 180 millimeters of mercury. 180 millimeters. Now at this point, what is the state of the heart? The state of the heart is that the right ventricle is completely filled with all this shown here is the blood. So 70 ml, 70 ml is filled now, and the pressure inside is 180 millimeters of mercury is the pressure that is built inside the ventricles. So the diastole is complete now. Now the heart is going to contract. Now the heart is going to contract. So when the contraction starts, when you draw also, you start from here. The systolic phase is going to start. See, up to this point you had the diastolic interventricular pressure, which is just shown here. Up to this was the diastolic phase, the filling phase was this. Now the systolic is going to start. So you, from where do you start? You start from here and draw backwards like this. That is the pressure variation that you're going to study. The heart is going to contract. That is during systole. Now in systole, we are going to study the same. Okay. So I've seen why I'm repeating this several times is because I have seen students who draw from here. The systolic phase also this draw from here, and when I see them drawing, I can make out that they have not understood the graph. The concept of the graph is not clear to those students who start drawing from here. I don't understand. Anyway, it should come from here. If you are thinking in terms of a working heart, the systole should start from here, and slowly it moves like this, and you can see, what you can see is an increase in pressure as the volume decreases. And you can see at 30 millimeters of 30 ml, and 30 ml of blood is inside the ventricles and the heart is contracting, it is able to generate tension maximum tension. The pressure inside would be 270 millimeters of mercury, and this is the place where maximum tension is produced by the heart. How will you measure the tension? The tension is measured by taking the distance between these two graphs. So during diastole, this was the tension. The pressure was only 30, and here it's 70. So at this point, you can see the maximum tension developed. If you want to take the whole tension developed in the heart, you can take the area of the graph. So that is the maximum tension developed. After this 30 mark, when the blood comes to blood, the amount of blood starts decreasing, the volume starts decreasing from 30 to 20. What you observe is that the pressure declines. The pressure declines sharply, very sharply. This is how you have to draw the graph. You have to start from the diastolic phase, and then you have to move towards the systolic phase. And from this graph, what you make out is that during the systolic phase, you can easily make out your Starling's law. When the volume was when the volume was 30 millimeters of mercury, you had the maximum pressure developed. The graph didn't move or didn't behave like this. It didn't behave like this. It didn't behave like this. That is, at 50 millimeters or 50 ml of blood when it was there, it didn't show the maximum contraction. It showed it at a particular place. That's the shape of this particular systolic pressure curve. If you see, it showed like this, right? At 30, it showed maximum. And after this, what happened? It decreased. So what was we referring to when we were saying the volume, the ml was increasing, the ml was decreasing? Actually, I was referring to the length of the muscle fiber, when I was saying the volume increased. Because if you have 30 ml, that means the length of the muscle fiber is stretched to a particular limit. If you have 70 millimeters, the length is increased to a particular limit. It further increased. And when you see the systolic graph, you can easily make out your law, which is the Frank-Starling's law. That is, you can see that until a particular volume, you can see an increase in the tension, and after that you can see a decrease in the tension. Now if you think in the opposite direction, also you can make out that the systolic interventricular pressure is also having this curve fashion, like this. Think like this. Let me rub this. Let me explain. Now you have a heart. Just think, we have an experimental heart with us, and we are filling different volumes in the heart, and we are making the heart to contract with that volume. Now in that case, this is not a working heart, this is an experimental heart. In that case, you can draw the graph from this direction, from this place, the initial part, to this place, like this. You fill 10 ml of blood in the experimental heart, and then you make the heart contract. You will have this pressure developed inside. Now you fill it to 20 millimeters, and you make the heart to contract. This will be the pressure that is developed to 40. Now you fill 30 ml onto the heart. Now it makes the maximum pressure during contraction. Now you again increase the length of the initial length of the muscle fiber by introducing more blood, that is 40 ml. But now you will see that the pressure that is developed inside is less. Now experimentally introduce 50 ml, add 10 ml more into the experimental heart. Now you make the heart contract, again you see the pressure comes down. So which was the point where you could observe the maximum pressure during contraction of the heart, that was 30 ml.
The time is
അപ്പോൾ
11
ഇങ്ങനെ
o'clock
ചിന്തിക്കുന്ന
p.m.
സമയത്ത് when you think in this manner, like an experimental heart, a study, we are doing an experimental physiology study on a frog's heart or any other heart, a mammalian heart, and if you're making these recordings and drawing a graph, now you can have the same graph obtained. It's not a working heart, it's an experimental heart. In that case you can draw in the from the right to left or left to right. It's up to you. So these are the two ways of dealing with the graph. But if you want to explain Frank Starling's law, you can explain both ways, the working heart also and the experimental heart also. You will see that at a particular volume, which introduces a particular length on the muscle fiber, you get the maximum tension that is developed, and that goes off as you increase the volume. It's actually going off as you decrease the volume. The initial length is less, that is also decreasing the tension. You increase the volume, you're increasing the length of the muscle fiber, that is also decreasing the tension. So this length at a particular volume, in this example it is 30 ml. So this length, you can call this as the optimal length. So initially we were referring to this length at the starting, before you actually brought any blood, and you saw in my previous slides as the resting length. We have a resting for a heart, an optimal length for the optimal length will produce the max tension, the maximum tension, max tension. I hope this concept is now clear for you, and this was one thing that we had to explain this class. So as the end diastolic volume increases, the tension developed also increases. The tension increases until it reaches maximum and then decreases. Same things as in different ways. Now the question is, the tension decreases at higher degree stretch. Why? It is due to disruption of the myocardial fibers. It is due to disruption of the myocardial fibers. Now coming to the last portion of this class. This is a very important concept, and this also comes as a physiological basis question for you for the undergraduates, and that is regarding the long refractory period of cardiac muscle. The question would be like this: how cardiac muscle is actually protected from tetanizing frequencies? What does it mean? It means, suppose we are experimentally introducing frequencies at very short intervals. At very short intervals means, if this is the first stimulus, the second stimulus is very close, third is very close in time, the time graph of time. Introducing stimulus at very near, very close to each other. If I am giving stimuli onto the heart, will the heart also respond to all these nearby stimuli, or will it not? Because if you consider your heart is a vital organ in your body, and if the heart starts responding to all the stimulus which are very close, then the heart would be contracting at a very faster rate, and it might also lead to arrhythmias in the heart. Irregular contractions can happen in the heart very so close. So there should be a minimum uh, safety period for the heart for the next impulses to come and actually cause contractions. So maybe evolutionarily, due to these reasons, there might be this long refractory period that has been formed in our hearts. So this is very clear if you compare the right side with the left side. What is here? What is here is as the electrical activity. What you see here is an electrical activity of skeletal muscle, and this is the mechanical event, mechanical contraction of your skeletal muscle, and this part is your cardiac muscle. This part is a cardiac muscle. What is the difference that you see is in the electrical activity as described earlier. This is a spike potential and this is a plateau potential. So this plateau potential, if I draw the absolute refractory period and the relative refractory period, and if I superimpose the mechanical event onto the electrical event, and if all these things, the electrical event, the mechanical event, and the depiction of the absolute refractory period and relative refractory period, if all these things are superimposed on a single graph, you have to draw this on your exams to show this interesting thing that the electrical activity is actually masking the mechanical activity, or the mechanical activity is actually protected inside the refractory period of the electrical activity. What does it mean? What does it mean? It means that no second stimulus will make a mechanical event possible in the heart, because it's lying the mechanical maximum. See, the maximum contraction is here. Until this point, the heart is actually contracting to its maximum. It's still contracting. Yeah. Now, at this point, contraction is almost coming down. It's almost finished the contraction, and because this is the relaxation part, this is the contraction part. So heart is almost relaxed, and see the electrical event. This part is the relative refractory period, but this part during contraction is absolutely refractory. That means it is in the absolute refractory period, and during contraction, a second stimulus applied, it will never respond. It will never respond. Mechanical event will never respond. But during the relaxation period, if you apply a supra-threshold stimulus, supra-threshold കൊടുത്തു കഴിഞ്ഞാൽ, then the heart will respond. How will the response be? If you apply a supra-threshold stimulus on the relative refractory period, I will show here. So like this. The first contraction, the mechanical event, the contraction is over, it's relaxing now. It's out of the relative refractory period. Sorry, of the absolute refractory period is here. Now it's out of the absolute refractory period. Now at any time, if you apply a second stimulus, see the second stimulus comes here, then it will contract like this. So it will be second contraction, and you also observe that the second contraction is having a higher amplitude than the first contraction. All these reasons for all these things would be learned in your experimental physiology practicals. You need something to learn in experimental physiology practicals. So there, in experimental physiology practicals, you will see the graphs, all those graphs in which of those graphs which are obtained on doing experiments on frog's heart, wherein you will learn all these concepts, why this is the second stimulus is higher than the first stimulus, and all these things you will learn in the experimental physiology. Now at this point of time, what you have to learn, or what you have to understand, is that the mechanical event is actually very safely enclosed in the electrical activity, because it's a plateau potential. And now you will be understanding why evolutionarily in the heart, in the cardiac muscles, there is calcium involved, which has led to a plateau potential, and that's actually a safety feature for the heart. I hope you understand the difference between the activities of the cardiac muscle and the skeletal muscle. Skeletal muscle, you see, this would be almost the point where your absolute refractory period would be. That this would be your relative refractory period almost. See, by this period, the muscle is still in its contraction phase. Still contracting, man, still contracting. So if you apply a suprathreshold stimulus in the initial part at around 5 milliseconds, if you give second stimulus, definitely it will add on to the first contraction. Add on to the first contraction. Definite, because it's in the relative refractory period of the skeletal muscle. So there addition can occur, superimposition can occur. All these things are possible in skeletal muscle, but cardiac muscle, that is possible. If it was like that, the heart would have contracted in a very weird manner, and for survival, that is not good. So this explains how the cardiac muscle is protected from tetanizing frequencies, or why, yeah, that's the same thing. How cardiac muscle is protected from tetanizing frequency, or what is the significance of having a plateau potential in the electrical activity of the cardiac muscle, or why there's calcium involved evolutionarily, how this changed, how the importance of calcium came in the contractile mechanism of cardiac muscle and smooth muscle. That is basically explained the same way. It's actually a protective mechanism. And that ends this long class. Almost, I think this is coming around 2 hours um, of your class on your cardiac muscle. Now in the next class, I'll come with your smooth muscle. Thank you.